Hello everyone and welcome to this audio podcast today. This audio podcast is part of the Serology and Squil. This audio podcast is part of our Hustle and Serotype. This audio podcast is as part of a Defcon premiere, and this excuse me, and this audio podcast is part of a Storm Lobostics production. So continuing on looking at immunobiology and stratifying receptors, and looking at and continuing on, because the goal of the one of the goal of the objectives of pharmacotherapeutics is in around disease manipulation with minimal impact on normal host immunology. The specificity offered by chemokines make extremely sort of appealing targets. Early research had informed that the survival of heterotropic cardiac allografts is prolonged in CCR1 negative mice, laboratory mice, and that lower doses of older generation IST cyclosporin A or CSA are required to completely protect the allograft in these mice. It has now in more recent work have been identified that similar phenotypes are detected in both CCR2 negative and CCR5 negative murine laboratory models. In addition, antibodies to CCR5 attenuate the development of graft versus host disease, GVHD, and also in a murine laboratory model. Research subsequently also looked at CCR5 to identify whether this chemokine affected survival of heterotropic cardiac allografts in CCR1 negative mice. And the investigations did not show whether this effect was due to neutralization of CCR5 depletion CCR5 expressing or depletion of CCR5 expressing cells, but did show that CXCR3 positive T cells and the CXCR3 ligand IP10 within endomyocardial biopsies is and was associated with AAR. Laboratory investigations had also identified that T helper cell type 1 dependent murine laboratory models of immune nephritis and severe combined immunodeficiency followed a sequence of invading inflammatory cells that might be initiated by antigenic specific T helper lymphocytes encountering antigen in the kidneys. This subsequently directs monocytes and possibly dendritic cells towards injured renal tissue. Early data had informed of several investigations in human experimental glomerulonephritis and that had shown an expression of T helper 1 related chemokine receptors CCR1, CCR2, and CCR5 in glomerular and mainly interstitial areas, as well as the urinary secretion MCP1, which had shown a close association to a disease activity and the phases or different phases of disease progression. The authors or investigators had informed in an inflammatory atmosphere interleukin 10 and blocked the downregulation of CCR1, CCR2, and CCR5 while uncoupling signaling activities. Thus, uncoupled receptors remain being expressed on the cell surface wherein they trap pro-inflammatory ligands and thereby serve as molecular sort of sinks and scavengers for inflammatory chemokines. Serve as molecular sinks and scavengers for inflammatory chemokines, removing them from the site of inflammation. Therefore, in early works, investigations had sort of informed that the induction of immunobiological decoys might serve as a tool for interfering with excessive leukocyte recruitment and activation. That draws this audio podcast to a close. This audio podcast is part of the Serology and Squil. This audio podcast is part of a Hustle and Stereotype. This audio podcast is part of a Defcon premiere. And this audio podcast is part of a Storm Robotics production.